Students, in our previous video, we have done the process of fertilization. Now we'll do the post-fertilization events. But let us have a recap of fertilization. We have done that one single ovum is surrounded by many sperms, and these sperms, they release the chemicals present in the acrosome. And this acrosome is having the enzyme. These enzymes, they help to dissolve corona radiata and the zona pellucida so that a single sperm can enter inside. Collectively, all these are known as acrosomal reaction. After this, when the one sperm has entered into the ovum, then there are cortical granules which are present below the plasma membrane of the ovum. They get fused with the plasma membrane and they are also releasing the enzyme. Now they are going to harden the zona pellucida so that no more sperm can enter inside the ovum. This process is called the monospermy. After this, the haploid male nucleus and the haploid female nucleus. When the sperm was entering into the ovum, then the ovum that was in the secondary oocyte stage got the stimulus to divide to complete the second meiotic division, and then it formed the ovum. So now this female nucleus and the male nucleus, they get fused and they form a diploid structure, zygote. This zygote is the beginning of a new life. All information of a new life, the blueprint of the new life, is present in this zygote in the form of chromosome from the male as well as from the female gamete. In this figure, the journey of this zygote is shown. Within the fallopian tube, this zygote start dividing. First of all, it divides into 2, then 4, then 8 cells. The initial division that occur in the zygote are known as cleavage. One more thing, students, human ovum is not having any yolk. Birds, they are having lots of yolk in their egg because the source of nutrition for the growing embryo is yolk. But in case of mammal, growing embryo, they will get nutrition from the mother through placenta. So there is no need of any yolk here. So when division is going on in the zygote, the whole cytoplasm get divided equally, and this type of the cleavage division is known as the holoblastic cleavage division. Remember, so in birds, this cleavage division is unequal. Some blastomere are small, some blastomere are large, but in case of mammal, all blastomere are equal. What are blastomere? The cells which are formed after the cleavage division. One more thing. Still, this zygote is surrounded by zona pellucida. And zona pellucida is very hard covering. We have done that the cortical granules, they combine with the plasma membrane with the zona pellucida, and this has already formed a very hard covering. So because of this hard covering, though the cytoplasm is dividing, though the number of cells are increasing, but the overall size is not increasing. Look at this figure. This is the initial zygote state. This zygote divides into 2 cells. Then there are 4 cells, and then multiply into a 32 cell stage that is called morula. But in all these stages, there is one thing that is common, is zona pellucida. So the overall size of blastula has not increased, though the number of cells have increased. What does it mean? That the nuclear content is increasing, but the cytoplasmic content is decreasing. In the next stage, or the blastocyst stage, morula is a mulberry-like structure. 8 to 16 or 32 cell stage is called morula. Student, there is one thing very special about morula is that all the cells of morula, they are totipotent. Totipotent means they are having the tendency to make any structure, also known as the stem cells. So many researches are going on these stem cells, There is no differentiation so far in this morula that this cell is going to form this structure, this cell is going to form this structure. No, there is no differentiation so far. But now this is going to enter into next differentiated stage. That is blastula or blastocyst. Still in this stage, zona pellucida is intact. Now the rearrangement of the cell start taking place, and now the cells get arranged into an outer layer that is called trophoblast. This is the outer layer that is called the trophoblast, and an inner cell mass, and a cavity appear here. This cavity is known as blastocyst. So student, in morula, there was no differentiation of the cell, there was no cavity, but here you can see that there is a cavity and there is an outer layer trophoblast and there is an inner cell mass. Now let us see the role of this trophoblast and the inner cell mass. Here you can see the trophoblast layer by the black outline, and this is the inner cell mass. Trophoblast cells, this is going to form extra embryonic layer. What do you mean by extra embryonic layer? Extra embryonic layer are those layers which are helping in protection, in nutrition, in placenta formation. They are directly not involved in any of the embryonic structure formation. On the other hand, inner cell mass, this is going to form the proper embryo. The inner cell mass, this first of all will divide into 3 germ layers. You know students that we are triploblastic. Triploblastic means we are made up of 3 germ layers, and these 3 germ layers are ectoderm, endoderm and mesoderm. Are all body organs, they are made up of these 3 organs only. There are lower organisms like coelenterata. These are diploblastic. They are just having 2 layers, ectoderm and endoderm. So here you can see the formation of ectoderm and endoderm layer, and in between these 2 layers, mesoderm is formed. The complete epithelium, nervous system, this is ectodermal in origin. They are formed from the ectoderm. Brain is formed from ectoderm. The complete alimentary canal, this is formed from endoderm. Heart, kidney, they are mesodermal in origin. So each and every organ in the body is having origin in these 3 layers only. Now after all these division, that is the formation of the morula, formation of the blastula, this occur within fallopian tube and the passage towards the uterus. The further division and the further differentiation of this blastula will occur only after implantation. What is implantation? This blastula, it moved down to the uterus, it get attached to the uterus. Now the uterine cell, the endometrium wall, the cell of the endometrium, they will multiply repeatedly. They will cover this blastocyst all around, and you can say they engulf this blastocyst inside. The blastocyst get embedded in the endometrium, and this process is known as implantation. I think this will be more clear to you now. This is the single cell structure zygote. And as it start moving in the fallopian tube, it start dividing. Within 30 hours of fertilization, this form two blastomere, and zona pellucida is very much intact. So now the four cells, a group of cells, zona pellucida, check it, it is still there. So this is a mulberry-like structure, all stem cells, no differentiation, but now a cavity start appearing. So this is blastula. And there is an outer trophoblast, and there is an inner cell mass, and still zona pellucida is there. Now just before implantation, zona pellucida is shed off. And now this blastula, it get attached to the endometrium. This stage is the late blastocyst stage.